Hey guys, in part one of this video, we already saw about the cell cycle and what happens at each stage of the cell cycle. We're going to focus now on mitosis and looking at those stages and what exactly happens. I want you to pay attention to the chromosomes and how they're moving, where they're separating. And so one thing that I didn't say if you watched part one of these videos is that at this point in mitosis, we know that the DNA has been duplicated. I'm going to use two colors. We'll do green and red. And we're going to say that the DNA, that it's green and red, is absolutely identical. Absolutely identical. But it's just different colors so that we can keep track of where it's going. So in prophase, which is the first step of mitosis, we know that we have double the amount of DNA at this point. So let's say that DNA, which is existing in a form called chromatin, which we'll talk later, which is DNA combined with associated proteins, is starting to coil up and condense in preparation for the cell to divide. So let's say that 46 of our chromosomes kind of look a little bit like this, starting to condense maybe a little bit. And then the identical other 46 looks the same, starting to condense into chromosomes. Now the nuclear envelope, which is the membrane that surrounds the nucleus, is also starting to break down at this stage because there can't be a nucleus. If the cell is going to divide, the nucleus needs to disappear so the DNA can freely move to either ends of the cells. So our nuclear envelope is starting to kind of break down a little bit outside of the chromatin that's condensing. In your models, you might have also included the centrioles, which are an organelle from which spindle fibers come, and the spindle fibers will help pull the chromosomes apart. So the centrioles are present. They kind of look like cylinders. There's two copies now. They're going to move to either end of the cell. So following prophase is metaphase. Metaphase is characterized by the chromosomes actually lining up at what we call the metaphase plate. The metaphase plate is like this imaginary equator of the cell. I'm going to adjust my camera. So the metaphase plate is kind of the center of the cell. Metaphase plate. The metaphase plate is where the chromosomes will line up, and now at this point our DNA looks very much like X's, and they're bonded at a point, this would be my wrist, called the centromere. And what happens is this side is absolutely identical to this side. So watch the way that I draw this in terms of color. So here's a chromosome. Here is its identical other side. Okay, same thing here. Here's another chromosome. I'll do two at a time. Here's its identical other side. My blue isn't working so well. Okay, the spindle fibers are attached at the centromeres, which are the centers of each one of the chromosomes. And they're extending from those centrioles. And they're going to pull the cell apart this way. And you'll notice that this red DNA is going to go to that cell, and this blue DNA is going to go to that cell, but it's the same DNA. I asked a question in class, if right here on this chromatid, if that is the gene for hair color, what is the gene on this side? It's opposite chromatid, and the answer would be also the gene for hair color, so that when the cell divides, both of them receive hair color. So after metaphase is anaphase. Anaphase is really cool. When we watched the real-time video, you could actually see anaphase happening when the chromosomes separate from each other. In anaphase, these spindle fibers kind of reel in, and literally the genetic, or not the genetic, the molecular mechanism is the proteins, these are proteins, get broken down, kind of like they're being reeled in, which is pretty cool. So it looks like this side of the cell is going to get the red copy of the DNA. This side of the cell will get the blue copy of the DNA. The spindle fibers are shortening. And we can tell the cell is starting to pinch in. The next phase after, this is anaphase, sorry, anaphase. The next phase after anaphase is telophase. Telophase and cytokinesis happen almost at the same time, and it's kind of hard to differentiate between the two because mitosis is not a step process, it's all continuous. So at this point, telophase is really marked by the cell starting to become two distinct cells but hasn't completely divided yet. At this point, something that is new is that the chromosomes are starting to decondense a little bit. Same thing on this cell. The nuclear envelope is starting to reform around the DNA. And mostly the spindle fibers have broken down. This is telophase. Cytokinesis is literally the separation of the cytoplasm, which is kind of happening at the same time here, but for the sake of modeling this in our class, we'll say at this point, if I were to draw the completion of cytokinesis, I would draw two completed cells that look a lot like they're in interphase, which means they're not dividing. Their nucleuses are fully formed. They have DNA in its spaghetti chromatin fashion. Same thing with this one. And this would be the end of cytokinesis. The key part that I want you to know is that this cell is absolutely identical to this cell. This cell will enter the cell cycle, so will this one. This one will divide, so will that one. We'll end up with more cells. 2 from that one, 2 from this one, we'll have 4 instead of 2, which came from originally just 1. So again, this is showing mitosis. Mitosis is in the M phase of this cell cycle. Cells will re-enter the cell cycle as many times during their lifetime until the cell ultimately dies. Later on, we're going to be talking about S phase and what happens during S phase, which is the duplication of DNA. But right now, we're talking about how cells themselves duplicate or how they divide, which is mitosis. So thanks for watching. I hope that was helpful. Ask me questions in class. Best of luck studying.